Okay, so I'm going to draw something out for you, and we're just going to go from there. What have I drawn? Everyone always says it's a mitochondria, but if you haven't picked up, my mitochondria have always been drawn this way. So I'm kind of doing something on the opposite side of the mitochondria. So what would you say then? You're actually still wrong. I'm drawing something inside the chloroplast, a thin thylakoid. So this would be a thylakoid. And if that's a thylakoid that I've drawn out right here, what would that make this entire sheet of paper to the outside? The stroma, which is within the what? So if I wanted to give a title of this picture, here's what I would title it. A chloroplast, which has inside of it thylakoids. I have drawn one out for you. And that would make this area, as we had said, the stroma to the outside. I've drawn it from left to right, showing you the order. So the thylakoid reactions have to be happening first. The stroma reactions happen second, which is kind of, if you start to think about it, a little bit of a reversal of cell respiration. Oh, and for the third time today, what are the two products of the thylakoid light reactions?
ATP and NADPH.
So ATP and NADPH, and we need to be thinking about that. I've got to get ATP and NADPH out of this system. And those two items are going to be used in my stroma's Calvin cycle. Okay, so let's start off with something called a photosystem. There are two photosystems that you have to know. Photosystem 2 and photosystem 1. Photosystem 2 occurs before photosystem 1. A lot of people always say, well then, why were they named as such? Because they were named in order of their discovery, not in order of their occurrence. Photosystem 2, again, happens before photosystem 1. And within each photosystem, you have pigment molecules. I've talked about several already, but what was the go-to pigment you've heard of all of biology? Chlorophyll, right? So I'm going to put my little chlorophyll pigment right here and right there. So that's the chlorophyll pigment. It is the housing unit of an electron, which I will put right here. Now, electrons, it's that little thing that we've been talking about over chapter 9, little negatively charged particle. Electrons can be excited. How do you excite an electron? You give it energy. It's like, you can imagine like a baseball. Unexcited in your hand, very excited as you throw it up. And how do you get excited? Well, the baseball gets excited by giving it energy. Now, ultimately, I had established where the energy is coming from, which is where? The sun, the photons from the sun. So I'm going to draw out a little sun. There's my little sun. It's got these little light beams, rays of energy. And let's view a photon, or I'll show you a photon, striking photosystem 2. Now, as the photon strikes the second photosystem, or photosystem 2, what will happen to the electron? It gets excited, which means it gets higher in energy. We depict that like such. Now the electron is in this excited state. There are a series of electron transport proteins located between your two photosystems. So some electron transport proteins. And what do you think they're going to transport? Okay. And to where do you think that these electron transport proteins will transport the electron? Probably to photosystem 1, which appears to be the next stop. So the electron is passed through those electron transport proteins and passed off to a chlorophyll pigment in photosystem 1. Now, a question to you that I might have is, if a photon can strike one time, do you suspect a photon could strike a second time? Okay. And of course the sun is shining pretty often, so it would make sense that a photon could strike maybe this first photosystem, or photosystem 1, which occurred second, striking the electron. Now, what do you think will happen to the electron once it is struck again with a photon? So it gets excited a second time. That means it's pushed to a higher energy level. And just like before, who was taking those electrons out of the energized state from the photosystem? An electron transport chain, right? So I suspect there will be a second electron transport chain that readily accepts the electron. I'll show you now. Lastly, that electron is passed off to a big enzyme over on the corner. So I'll put this massive enzyme over here. And the electron is passed off this direction. So we have to be thinking to ourselves that over the course of time, electrons would begin to build up at this location. I don't really have anything of appropriate size, so I'm going to be using a piece of orbit gum. Let's say that this orbit gum is the electron. The electron was excited to that stage of photosystem 2, passed on through an electron transport chain to photosystem 1, excited again, and dumped off over on this enzyme. So the orbit gum, or the electron, which it represents, was here initially and ends up over here. Agreed? Did it take energy to move that electron, or that orbit gum? So in the case of the orbit gum, my fingers supplied the energy, but in the case of the electron, ultimately where did that energy come from? Okay, from the sun, the light, the photons. And if the electron was initially there, you could definitely say that at the end, it is located on this enzyme way over here, correct? So at the end of time, not at the end of time, but after this process has occurred, tell me about this spot right there. Is it electron heavy or electron less? Because the electron was moved to way over there. Now, it's pretty obvious that if an electron gets moved this direction, a new electron is going to have to fill that void, correct? What was the final electron acceptor in respiration? Which helped to form what molecule? Water. So you could say that water was the final product of cell respiration, right? Ironically, water is the first reactant of photosynthesis. So if water is a reactant, I think it would be broken down. Now, what gas do you know photosynthesis produces? Oxygen, right? So definitely O2 has to be a product of this. But why break down the water? What does it have inside of it that I need? Especially at this point right there now. Electrons, right? So I'm going to produce some electrons. Now, when oxygen was the final electron acceptor, it gained the electrons. So there's the oxygen and the electrons that it gained. And what else did it combine with to form the water? And it was just H+, right? So I bet the H+ is going to be formed as well. I'm not going to really worry about balancing this reaction at all. I just want to see the products in a qualitative fashion. Now, a couple of questions or points to make. The first point is, where does this oxygen go? What's that used for? I can think of two areas. This oxygen could be used in that plant cell's mitochondria, or it could just be released from the plant, because you know plants are producing oxygen, right? Let's assume the latter. So we'll say that the O2 goes out of the plant, and it goes through the little openings through which gas exchange occur. That's called the stomata. What about the electrons? Where do they go? Kind of a no-brainer. There was a reason that we had to do this reaction. I think the electron has to go to PS2. You have to replace the electron that got bounced and moved around. So this electron, in a way, is a replacement mechanism. Or you could say a replenishment mechanism, whichever you prefer.